right. Good morning, everyone. Thank you for joining BrainMap today. Before we start, I want to remind you that this BrainMap Seminar Series is co-sponsored by the P41-funded Center for Mesoscale Mapping housed in the Martinos Center. It is my pleasure to introduce our today's speaker, Dr. Jelle Veraart. Dr. Veraart is an assistant professor of radiology at New York University, and his research is focused on the integration of advanced image analysis, preclinical validation, and clinical translation to study neurodevelopmental and psychiatric disorders, non-invasively and at the microstructural level. He has extensively worked in diffusion MRI to drive forward treatment and understanding of brain mechanisms by enabling microstructural specificity through modeling and multidimensional MRI acquisitions. Today, he will talk about on the quantification of axon diameter distributions using diffusion MRI. Jelle, the virtual stage is all yours.
Thank you so much for this introduction, and good morning, everyone. Thank you for to the organizers for inviting me to present my work on axon diameter mapping using diffusion MRI in this series. It's a true pleasure and honor to present this work to you, especially because most of this work has only been possible because of technical developments that were made by you and your colleagues. And I think today we do share the interest in exploiting ultra-strong diffusion-weighted gradients, microstructural imaging, and axon diameter mapping. So I hope that this talk will be the starting point of some interesting discussions as well. Today I will break down my talk in three parts. The first one is how can we improve the specificity of diffusion MRI to axons by exploiting strong diffusion-weighted gradients. This is a little bit more of an introduction. Thereafter, I will follow with the bulk of this talk, which is axon diameter mapping using diffusion MRI, including topics of the theory, the validation, and human translation. And to conclude this talk, I will address clinical application, which focuses on children with 20q11 deletion syndrome, and we try to detect changes of axonal morphology using this novel type of biomarkers. Now, amongst all neuroimaging modalities, diffusion-weighted MRI stands out for its sensitivity to microstructural changes associated with development, aging, and disease. And therefore, over the past 2 decades, diffusion MRI became a common modality in clinical scan protocols. And the most successful clinical applications so far have been detection of stroke lesions, pre-surgical planning. However, there is lots of ongoing research to further promote diffusion-weighted MRI as a clinical and research tool. Unfortunately, the brain is vulnerable to many disorders and diseases. And those disorders and diseases comes with like a wide variability of pathological and cellular processes. Inflammation, gliosis, cell swelling, axonal degeneration, and so on. Diffusion-weighted MRI is not specific to those processes. Diffusion-weighted MRI is generally not specific to a particular cell type, and that's because if you zoom into one particular voxel, we actually see that it's like an extremely rich and diverse and complex microstructure underlying that imaging voxel. So if you do see a change in a diffusion-weighted signal, it's not necessarily clear or obvious which particular cell or which particular change drove this signal change. But I do believe that we are ready to break this paradigm. But to do so, we have to have a very good understanding of what we're measuring, how the microstructure is encoded in our diffusion-weighted MR signal. And for people who are not familiar with diffusion-weighted MRI, the following slides will be extremely important to you. If you think about diffusion-weighted imaging, one metric that comes to mind is the mean diffusivity. We often report the mean diffusivity as a biomarker for microstructural changes. Mean diffusivity is easily associated with the diffusion coefficient of water in, for example, the brain. But that's incorrect. The diffusion coefficient of a fluid is a physical property that depends on the viscosity and the temperature, not the microstructure. What we are measuring with diffusion-weighted MRI, what we are reporting with diffusion-weighted MRI is the apparent diffusion coefficient, and that's an MR property. It's an MR property that depends on the viscosity and temperature, but it also depends on the microstructure of it and in the diffusion-weighted encoding scheme. What we measure with diffusion-weighted MRI, it's basically the dephasing of spins that are a consequence of their displacement. And the mean square displacement of a freely diffusing particle is proportional to two things, the diffusion coefficient and the diffusion time. That basically means that if you give a particle more time, it will on average move further and further and further from its starting point. And that's a physical property. However, if we start considering a particle that is hindered or restricted in its movement by, for example, the microstructure, cell membranes, boundaries, and so on, then we might see that this displacement becomes constrained, and therefore we might encode sensitivity to those presence of those membranes. But that only happens if the diffusion length of a freely diffusing particle exceeds the cellular dimensions. And we can do some very simple math to demonstrate that that's the case in the human brain. The diffusion length depends on diffusivity and diffusion time. If you plug in some typical values, and we know that in our brain, if there would not be any cells, if there wouldn't be any boundaries, that a particle would want to travel on average about 20 micrometers. And in comparison, the axon diameter is about 1 micrometer. So we've got like an order of magnitude difference. And that's something that we can exploit to increase the specificity of our diffusion-weighted signal. The diffusion length, basically the mean squared displacement, is proportional to diffusion time and diffusivity. Now, if that diffusion length is no longer a dependent variable, but becomes a constraint by, for example, the cell dimension A, then we can reshuffle this equation and we can write basically the D, the diffusion coefficient, as a function of this cell dimension. But that number is smaller or equal to the actual diffusivity. And therefore, we have to introduce this apparent diffusion coefficient. And the apparent diffusion coefficient is now proportional to the mean squared displacement, and it encodes the microstructure because it encodes the cell dimension. And that's a very important difference between the apparent diffusion coefficient and the diffusion coefficient. It's because we've got a constraint, hindered or restricted diffusion, that there's like a difference between them. It is exactly that we're measuring. If we employ a pulse gradient sequence, Jessica-Tannert sequence, we basically introduce a dephasing and rephasing of our spins, and there's like a separation of that event, which is a diffusion time. Any imperfect rephasing will introduce a signal decay, and that's the contrast that we use to quantify our processes. A rough approximation tells us that our diffusion-weighted signal decays exponentially fast, depending on a b-value, which is kind of like a convoluted metric quantifying gradient strength, duration, separation. It's kind of like our diffusion weighting and coding strength, and apparent diffusion coefficient. But the main message here is that the further a proton can move, the larger the apparent diffusion coefficient, the stronger the signal decay. And that's what we're going to project on our microstructure. On the left-hand side, you see the complex microstructure of the white matter. It's just confocal microscopy, and 3 cell types are labeled. We've got axons, cell bodies, and astrocytes. Building on some assumptions, building upon some prior knowledge, we can make a sketch of this complex environment. And within the diffusion community, we present this model of axons embedded in an effective extra-axonal space, which is shown on the right-hand side. In such an environment, we have different compartments. If you consider a proton, which moves in the extra-axonal space, which is the green curve over there, your movement is hindered by the presence of axons. You can't move as far and as fast as free diffusion, but if you're giving some time, you can still travel great distances. A proton in the intra-axonal space, a proton which is trapped inside of the axons, which is assumed to be impermeable for relevant diffusion times, that's not the case. Your mean squared displacement is bound. It is bound to a certain value, which depends on your diameter, and it's independent of the diffusion time after a while. Now, if you look to the diffusion-weighted signal decay inside such an axon, and we mainly look at the perpendicular diffusion, then we now know that apparent diffusion coefficient depends on the radius of the axon. And the radius is A over here. A typical radius of an axon is about 1 micrometer. And the typical diffusion time is about 50 milliseconds. So apparent diffusion coefficient inside such an axon is 2 orders of magnitude smaller than the average apparent diffusion coefficient in a normal white matter voxel. And that's a very big difference. And for many practical purposes, one concluded that the diffusion inside of the axon is 0, can be ignored for all practical purposes. And that is something that brings us to the main message of the talk. We've got the same image of diffusion inside of axons versus a diffusion outside of our axons. And we know that we decay exponentially fast, but that our apparent diffusion coefficient is different and depends on the compartment where we're at. If we are in the extraxonal space where hindered diffusion dominates our signal, we're exponentially fast and we've got a relatively strong apparent diffusion coefficient. Empirically, we think in terms of 1 micrometer squared per millisecond. In the axons, which are trapped for our diffusion times, our ADC is assumed to be 0 or very close to 0. That means if we even go to extremely high B values, we don't have a strong signal decay. We don't have a signal contrast inside of the axon. The contrast comes from the extraxonal space. And if you go from the signal decay to a signal fraction plot as a function of B value, then you see that at clinical B values, and we think about B value of 1 second per millimeter squared, we have significant contributions of both the intra and extraaxonal compartments. However, if you go to stronger B values, and then there are B values which are kind of like underexplored in our community, the intraaxonal space starts dominating the diffusion-weighted signal because the extraaxonal space decays much, much faster, exponentially fast. So at high B values, there's no residual signal coming from the extraaxonal space. We empirically concluded that B6000 is kind of like a regime where axons are extremely dominant to an extent that the extraaxonal space can be ignored. And that brings us to the point that the diffusion-weighted MRI can become like a filter for axons. And that idea is not new. I mean, we often start off with a picture of myelinated axons inside of an extraaxonal space. And within our community, it's very well adopted that by going to long echo times needed to accommodate diffusion-weighted gradients, we suppress the myelin signal. So we kind of like filter out myelin already. And I just add like one more filter. By going to high B, we remove the extraaxonal space. So where we start with myelinated axons in extraaxonal space, by going to long echo times and high B values, we suddenly only have our intraaxonal signal. It is orientational dependent, but by doing some powder averaging, we can factor out the dispersion and we basically virtually align our axons. And this is something much easier to work with. I just plot the same image, the same subject. We acquired about 30 diffusion-weighted images at different B values, 1000, 2500, and 6000. And at the B1000, it's actually very difficult to see contrast even between the gray and the white matter. We go to B2500. We start seeing stronger contrast. We see stronger contrast between the white and the gray matter. We see a little bit more structure in the white matter itself. But if you go to B6000, you can see how clearly this map highlights the white matter with very strong contrast. And this is kind of like getting some feel on the importance of using B values as a filter of axons. And this is not a new idea. Work within your group already showed that by going to high B values, you can get like much sharper fiber orientation distribution functions than at low B value. And it's my understanding that this is in part because the extraaxonal space is much less is much more dispersed, it's much less coherent, and therefore you kind of have a blurring of your orientation distribution function. So by going to the high B values, where you're more specific to the axons, you get just sharper fiber orientation distribution functions, which is, of course, is helpful for fiber tractography. Just some streamlines that I computed using publicly available software. B1000 versus B10000, and you can see that the segmentation of the cortical spinal tract becomes much easier with less spurious fibers, in part because of the sharpness of those fiber orientation distribution functions, and mainly because of the extraaxonal space not blurring the ODF. Now, if you go to the high B regime, we can start quantifying features of axonal morphology, densities, volume fractions, and hopefully also radii. And that's what this part of the talk will focus on. The white matter of the axons have like a very interesting role because they affect the rate and the speed of the conduction of our brain signaling, and therefore there's like a direct link between brain structure and the brain function. Being able to probe or to measure an altered axonal diameter distribution, which has, for example, already been observed in neurodevelopmental disorders and pathology, we would create a biomarker to study the brain function in vivo, non-invasively, and globally. And that would definitely promote an early and more accurate diagnosis of various brain disorders. But we do keep facing this challenge that we are trying to probe something on the sub-micrometer scale. And that caused quite some controversy in the field. Axon diameter mapping using diffusion MRI has been investigated for about 15 years now. And there's what we call a notorious overestimation. Histology in human white matter shows that axons have a radii of about 0.5 to 2 micrometer with only like a few axons exceeding those numbers. With MR, we have much larger values. We go to 3.5 to 15 micrometers. Lots of work has been done on trying to understand confounding factors, modeling assumptions, for example, assumptions on non-Gaussianity in extraaxonal space has been addressed, dispersion, and so on. And on the other hand, one has questioned histological preparation and how it might contribute to this mismatch between histology and MR axon diameter mapping. But we're here to try to kind of contribute to this controversy and try to resolve the debate, or at least like try to make a constructive contribution to this debate. And then we go back to this previous slide. In this previous slide, I showed that the diffusion weighted signal perpendicular to the axon is basically equal to its non-diffusion weighted signal. So basically the apparent diffusion coefficient is assumed to be 0. But that's not entirely correct. Almost 0 is not 0. So if there is actually contrast, we might be able to measure it. But what do we need for this? And that's where gradient tricks comes into play, where hardware development comes into play. If we want to be able to measure axon diameters, we need to create contrast to noise and achieve a good contrast to noise ratio by maximizing B, the B values, times the intraaxonal diffusivity, because only then this exponent will be smaller than 1, and we can be hopeful to measure it. The B value is a known feature in diffusion MRI, which is basically a convoluted metric that includes the diffusion gradient strength, the diffusion gradient duration, and the separation, which relates to the diffusion time. The intraaxonal diffusivity perpendicular to the axon is actually a feature that has been studied in depth as a function of diffusion times in various regimes. But in the regime that's most relevant to us, Neumann did actually quite some work in the 70s already, showing that the diffusivity relates to the radius to the power of 4 and diffusion time. So if you make the product of those two variables, we actually get like a rather simple equation that shows that if we want to be sensitive to axon diameters, we have to maximize this number. And that means that we need large axons, because large axons, R to the fourth, drive the signal contrast. We also see that our signal will be proportional to the gradient strength squared. So we need strong gradients to provide contrast. What we also see is that there's like a linear relationship between the signal contrast and the gradient duration. What we don't see is diffusion time. So if we want to measure axon diameters using a pulse gradient sequence, we rely on strong gradients. We are limited to measuring large axons. And that's kind of like shown over here. I did some simulations on like how sensitive are we to axon diameters by plotting the interaxonal diffusivity versus the radius for different scanners. A clinical scanner, 80 millitesla per meter, the human connectome scanner, 300 millitesla per meter, and preclinical systems like with gradient strengths of 1,500 millitesla per meter or even higher. In a clinical scan regime, where the gradient strengths are limited to about 80 millitesla per meter maximally, we can only measure axons that exceeds more than 3 micrometers in radius. If you plot that, if you compare that to the typical distributions that we see from histology, there are like barely any axons in that regime. So we're not sensitive to it. If we go to the human connectome scanner, the connectome 3D scanner, then we can see that our resolution limit drops from over 3 to 1.4 micrometer. On the same distribution as this blue line, we start becoming sensitive to this tail. If we can go to preclinical scanners, this limit even decreases, and we can start measuring sub-micrometer. And then we really start like becoming highly sensitive to changes here. So even if you go to the strong diffusion-weighted gradients, as for example to the Connectome 3T scanner, MR axon diameter mapping will always be dominated by the largest axons. It is this tail that we are probing. And that brings us to this image. This is our voxel. We have all kinds of axons with different radii, like a complex distribution, as shown before. And this is what MR does. MR is a filter for large axons because of two reasons. The larger axons have most likely more protons, so they weigh in more strongly in a diffusion-weighted signal. In the previous equation, I showed that there's like a relationship to the apparent diffusion coefficient and R to the fourth, and that adds to the dominance of large axons in our signal. And now, of course, we are not measuring the distribution. We have to integrate all the axons within our voxel, so we get like one single number, which is what we call the effective radius. And that number represents the entire distribution that is dominated by the large axons. To be more specific, the effective MR radius is basically the ratio between the sixth order moment of the distribution divided by the second order moment of this distribution and then normalized. Sixth order moment, very tail heavy, so we're dominated by large axons. A question I often get is like, well, if we can measure this effective radius, can we inverse this problem and extract the average radius? Because average radius is something that we get from histology, and we would love to compare histology and MR. So can we map the effective radius back on the average radius? And the answer is no. I generated 3 distributions. They're all gamma distributions with like different form parameters. And what's interesting about those 3 distributions, which are clearly different, is that the average radius varies strongly from about 0.6 to more than 1.2. However, if we compute the effective radius by dividing the sixth order moment and second order moment, we can see that we kind of like end up at the same value. So we cannot go back from the effective radius to the average radius without knowing the exact shape of our distribution, of axon diameter distributions, which is unfortunately not known. One concept that I have to highlight a little bit more before we can move to the actual model of axon diameter mapping. Our diffusion weighted signal is a convolution between the fiber orientation distribution function and a signal kernel. The fiber ODF is typically used for tractography because it shows basically the main directions of diffusion, and we can integrate along those directions to reconstruct our streamlines. The signal kernel is basically a fiber bundle average signal response, which is rotationally invariant. And we can use that particular kernel to obtain all kinds of biomarkers, fiber density, diffusivities, volume fractions, axon radius, and so on. Estimating simultaneously the ODF in the signal kernel is like a highly complex landscape of optimization landscapes. So it would be much easier if we can cancel out ODF, that we can decompose the problem and focus on the signal kernel itself. And that's actually made possible by something which is called powder averaging. Our diffusion weighted signal is acquired along many, many directions. Simply averaging the signals per B value over all those gradient directions cancels out that orientation distribution function. The top image here is just a diffusion weighted image as particular gradient directions. The bottom image is the powder average signal. If we make a scatter plot between the B value and our diffusion weighted signal, we can see that per B value, we have like a big spread. And that spread relates to anisotropy, dispersion, and the fiber ODF. However, we can just average that, and we get like a simple signal decay as a function of the B value. And that simplification actually has big implications, because what happened by doing powder averaging, what happened by averaging all the signal across different gradient directions per B value is what you can call a virtual alignment of all the axons. So where the axons were initially dispersed with potentially different directions, they just kind of like virtually align. And that makes it so much easier to model this. If we have to model the powder average signal inside of the axon, we actually only need 3 parameters. And this model is actually very old. It's established by Callaghan in the 70s. That basically says that our powder average signals decays as 1 over square root of the B value multiplied by a correction term that depends on the radial diffusivity inside of the cylinder, the axon, and therefore depends on the radius. We've got this intraaxonal signal fraction F. We've got an intraaxonal axial diffusivity, and both combined, they result into like a prefactor beta. But our focus is on relating this intraaxonal radial diffusivity to the radius. Now, this model assumes there is no other compartments contributing to the signal. That means we are assuming that there is no contribution of the extraaxonal space. That means that we have to go to high B values. So this model only applies to high B values of 6,000 or more. But the good thing is there's no degeneracy in terms of the radius, and there's like a reduced number of assumptions and confounding factors because we don't have to model the extraaxonal space. The problems of the model, or the limitations of the model, we need 2 very high B values with a minimum of B6000 to solve it. We will always have to deal with like a low signal to noise ratio, and there's like a low contrast to noise ratio, and strong gradients are really required. And even if we can deal with those 3, we only measure the effective MR radius, which is heavily biased towards the largest axons in the voxels, which might limit its application. Now, that effective MR radius hopefully can become like a biomarker, but a biomarker has to meet several conditions. It needs to be accurate, precise, reproducible. So we started looking into like the accuracy and the precision of this metric. The accuracy has been assessed in a preclinical study in collaboration with Noam Shemesh and Daniel Nunes of the Champollion Foundation in Lisbon. And there we scanned perfusion fixed rat brains, 3 of them. And we focused on the mid-sagittal plane, focused on the corpus callosum. And just over here on the 3 maps, I show basically the effective MR radius along the corpus callosum, and you can see this beautiful low, high, low trend that is well known from literature in rodents, but also in humans. So that's kind of like a qualitative assessment of accuracy or gaining confidence in this technique. A more quantitative analysis was followed. We focused on the GNU of the corpus callosum, and we identified 16 patches. And 1 patch had the same size as an imaging voxel, which was about 100 micrometer by 100 micrometer. Within each patch, we did staining, axons, cell bodies, astrocytes, but we focused on the axons. And we quantified the radii of those axons. So within every patch, we can make a distribution. And from a distribution, you can measure the average, and that's what we would typically report in an histological analysis. We can do this for 16 patches, so we can make a box plot of that. And you see this kind of like a precise box plot over here. Now, we know that with MR, we're not measuring the average radius. So in order to facilitate a more honest comparison between MR and histology, the first step was to compute these effective radius that correspond to our histological data. So we computed the 6th order moment of every distribution, the 2nd order moment of the distribution, we computed the ratio, we reported this value. And we can see that our apparent radius increases quite a bit. By stepping from the average to the effective radius, we almost like double our nominal value. This brain and 2 other brains, we went to the MR scanner. We have kind of like location matched voxels. So we get like an MR value of the effective MR radius in every voxel. So we can make a similar box plot. And what you can see is that the values that we compute for MR are actually in good agreement with the effective radius that was predicted from histology. There's like a small remaining offset, which can relate to various confounding factors. But the main take-home message is that interpretation is important. When you compare modalities, it's important to consider what is encoded in those modalities. While microscopy, confocal and electron microscopy often focuses on the average values of a distribution, we can't measure that. We measure something which is still weighted, which is the effective radius. If we compare our results to previous models, we can see that there's like a strong reduction of the overestimation because we have less modeling assumptions. We don't have to deal with the extra axonal space, and that improves our precision, our accuracy. However, the residual overestimation, that is about 5 to 15%, remains questionable, and probably many other confounding factors needs to be addressed to explain this. Precision. We don't want this to be limited to preclinical MR only. We want this to work in human MR. And with the development of strong gradient systems in human MR, the connectome system in particular, there was an opportunity to test this. At that time, I was in Europe, so that's like a collaboration with the 2 European sites that have the Siemens connectome scanner, which is Kubrick of Cardiff University, and the Max Planck Institute in Leipzig. There were 5 healthy volunteers enrolled in this study, and 1 subject was scanned on both scanners. However, every subject was scanned twice. There was a test scan, a break, and a retest scan. And within every scan session, we acquired 60 B6000 images and 120 B30,000 images. There were 2 repetitions per session, and there were 2 sessions per subject. And the total acquisition time of the test and the retest scan was 24 minutes. And just to show a little bit of like image quality, single diffusion weighted image at B30,000 is shown on the left-hand side. The powder average signal at B30,000 is shown on the right-hand side. And I hope you do appreciate that this signal and its contrast to noise ratio is quite appealing and impressive. And it really shows that there's a little signal decay inside of the axon. You can really go to very high B values without losing signal and without hitting the noise floor. I just show the effective MR radius for the test and the retest data. Just qualitative assessment first. What we do notice is like a very good agreement. We see spatial variability that seems to be reproducible. And that, I mean, that made us optimistic that we actually have like a useful biomarker here. But we had to be more quantitative. So we extracted different fiber tracts, corporal spinal tracts, arcuate fascicle, and so on. And we plotted the tract averaged effective MR radius per subject, test, retest, and then also the one in Leipzig. And what you observed is that the inter-tract variability, for example, the difference between the CST and the arcuate, is larger than the intra and the inter-subject variability. And we also noticed that certain tracts, like the cortical spinal tract, have larger radii than other tracts, and that's in agreement with histology. So again, kind of like an optimistic message in terms of the precision, but also the accuracy of this technique. And we can quantify this even more. The voxel-wise test-retest variability is about 10%. It varies from tract to tract, but it's about 10%. And the inter-scanner variability was similar to the intra-scanner variability. However, 10% might not be sufficient for many applications. So we tried to work on the analysis side to improve this. And we were thinking that the voxel is kind of like an arbitrary imaging unit. If you think about axons, we think about fiber tracts. So what we can do is like we can compute tract segments rather than a voxel, and to quantify the axon diameter distribution. And if we move from voxels to tract segments, we basically include like a disk of the full bundle rather than a single voxel. We can improve the reproducibility quite a bit, by even more than a fourfold, and then we get like just like 2-3% test-retest variability, which is definitely sufficient for clinical research. We can do this whole brain. So we extract all the different fiber bundles, we make segments along those bundles, and we just basically plot the values along those tracts. And the axon diameter mapping shows a higher reproducibility in the along tract variability. And the along tract variability is again bigger than the within and the between subject variability. So we are sensitive to changes within our own brains. Can do the same thing in the corpus callosum, and then we can see that kind of like lower values in the GNU, uptake in the body, not as much a drop in the splenium as we would have hypothesized. And it gives us some thoughts about MR axon diameter mapping, both in terms of accuracy and the precision. There's like a reasonable accuracy in rodents. There's like a 15 to 20% like overestimation in the worst case, but there are a couple of assumptions and confounding factors, curvature, undulations, and lots of research and thoughts and on that we should definitely consider when interpreting axon diameters. And there's also an unknown intrinsic diffusivity that has to be chosen, but I'm not sure whether there's that much prior knowledge on that, to be honest. Overall, reasonable accuracy, acceptable reproducibility, even in human MR, and we have sensitivity to along and intertract variabilities. But if you just go back, we still have very high values of effective axon radii in humans, and especially in some tracts like the CST, but also part of the corpus callosum. And some of those along tract variabilities just doesn't make sense all the time. So I keep wondering about how specific are we to those axons after all? Because if you look at the CST, and I mean, it's a little bit of like an unfortunate windowing, of course, but we do see a significant drop if we go from bottom to top of the brain. We see a significant drop of our axon diameter. And that's not necessarily something that we expected. However, if we go to some of to the literature, and literature is focused on MR spectroscopy. There's like one paper that I found that it's MR spectroscopy along the same fiber bundle, so along the CST, and they plotted neuronal and glial metabolites, basically just concentrations. And be aware of the flip position here. NAA, which is neuronal, we don't necessarily like monotonous increase. We see variation along the tract, but it's like not monotonically increasing. While if you look at the more glial, the choline, which is more glial forward, you can see the same increase, which is kind of interesting. Diffusion-weighted spectroscopy has been studied as well. Axon of the measurement of radii of neuronal and glial processes have been done by applying a similar model to the metabolites using diffusion-weighted spectroscopy. And the top two, NAA and glutamate, are more axon specific. And then we can see that we have lower radii then we compare them to more glial forward metabolites, being like choline and myoinositol. And that makes us wonder, do those astrocytes weigh in our measurements? And I'm sorry, I have to go back for a second to show one image, but this is our confocal microscopy. Over here, we do have, in red, we've got those axons, micrometer thin. In green, we've got astrocytes, one of the dominant glial types. And you can see how the process of this astrocyte is massive, up to 7 micrometer in radius compared to axon diameter. This is like some food for thought. Keep being puzzled by these large values, and I do wonder whether there's like an important confounding factors being glial processes that might contribute to this observation. Now, can we move towards clinical application? Yes, to some extent. And this is a study that has been done in collaboration with Kubrick of Cardiff University. It was a study led by Dr. Erica Raven and supervised by Professor Derek Jones. And they were studying neurodevelopment and typical development and atypical development. And the atypical development was focused on 20q11 deletion syndrome. It's the most common microdeletion, and it's associated with cardiac defects, craniofacial abnormalities, and intellectual disability. It is a very high risk for autism spectrum disorder and other psychiatric disorders. So it's like extremely interesting to study this disorder, to understand the neurobiology that might be underlying various psychiatric disorders. And 20q11 has been studied in preclinical setup, and what we have observed, of what others observed, was like a functional hypoconnectivity within large-scale brain networks. And evidence from mouse model shows that there's like a disruption of cortical neurons. And the outstanding question is whether this hypoconnectivity in children with 20q11 deletion syndrome might be associated with changes in axonal morphology. And that hypothesis is supported by evidence from other disorders, being autism spectrum disorder and Angelman syndrome, which is another genetic disorder, where we can see that there's like a loss of large axons in both of them. And there's substantially difference, because in autism spectrum disorder, we have a loss of density, while the density is still very high in Angelman syndrome, but they share in common that there's like a loss of large axons. So this study included typically developing children, 92 in total, aged between 8 and 18. And there were like 6 children with 20q11 deletion syndrome. This study was interrupted by COVID, and we're hopeful that we can increase this number. But overall, there are like 14 age and sex matched typically developing children to compare to the 6 20q11 deletion syndrome carrying children. And the subjects were all scanned on the Siemens quantum scanner, exploiting the full 300 millitesla per meter. It's a multi-shell diffusion weighted imaging protocol, including low and high B values. And the highest B value is 6,000, and it's only one of them, so we cannot do quantitative MR axon diameter mapping. But because we are working on the quantum scanner, exploring the 300 millitesla per meter gradients, we know that this B6000 is sensitive to axon diameters. If we compare the signal decay as a function of the radius for the acquisition that was being used, basically B6000 with 300 millitesla per meter, we can see quite like a significant drop in our signal. So we've got a good contrast that we can assume there is like some sensitivity to axon diameters. If you would have done this on the Prisma of basically a clinical system with 80 millitesla per meter, we would not have the same sensitivity, and at lower B values, we would not have gotten it at all. So although it's only one B value, we will not be able to quantify the axon radius, but we are sensitive to changes in morphology. And that has been exploited here. Large-scale studies has observed that children with 20q11 deletion syndrome have a loss in white matter volume. And that loss has been reproduced in this study. We do see global brain, we do see like a whole white matter volume fraction decrease, and we also see it in individual tracts. But the question is like, what drives, or what mechanism causes this loss in white matter? Because we have got a multi-shell acquisition, we can do many diffusion studies. We can do DTI, DKI, we can do spherical averaging at different B values. We can do biophysical modeling using, for example, the standard model. And we can just look at the percentage difference between 20q11 DS and age and sex matched controls. And what we notice most is that there's like an increased sensitivity at higher B value. And that makes us believe that the microstructural changes that are associated with axonal packing and morphology are key features to be studied. We know that we are sensitive to the axon diameters. And we know that an increase in diffusion-weighted signal at high B values is in agreement with the loss of large axons. And we also see that our biophysical modeling, in this case, the standard model, reveal an increased density. But it only explains the difference partially. And the combination of strong signal decay at high B, an impartial explanation of our observation by density only, and the overall loss in white matter volume makes us conclude at this stage that there's like a disproportionate loss of large axons in 20q11 deletion syndrome in agreement with other neurodevelopmental disorders such as autism spectrum disorder and Angelman syndrome and Fragile X syndrome as well, actually. So we're hopeful that we are achieving a sensitivity to axonal morphology that is actually translatable to clinical studies. Even studies including children for which long scan times are really like problematic. So as a conclusion of this talk, strong diffusion weighted imaging filters out the intraxonal signal. So we can improve the specificity of our diffusion weighted signal by going to high Bs if we exploit strong gradients. The accuracy of MR axon diameter mapping has been promoted by the suppression of axonal dispersion using the powder averaging and the extraaxonal space by going to high B values. Regardless of our developments, there's an intrinsic bias of MR that have axon diameters to larger axons. We cannot replace histology. We cannot obtain a full distribution or measuring the average radius. No, we are measuring that tail of a distribution. But human translation is possible within acceptable scan times, but it depends on strong gradients. So developments of the quantum scanners and future developments will definitely keep promoting the development of these kind of biomarkers. And a pilot study on the use of this type of biomarkers showed basically sensitivity to changes in axonal morphology in a cohort of children with 20q11 deletion syndrome. And by that, I would like to conclude my talk. Definitely many people to acknowledge here, because this work has been conducted over multiple years in collaboration with different institutes. I was lucky to spend time in NYU and the Champollion Foundation to do the theoretical foundation and the preclinical validation. And I was very lucky to have been able to spend some time in Kubrick to work on the reproducibility. So everyone included in this project is yeah, I'm very grateful for all their contributions. Okay, so this is my talk, and yeah, I would love to hear your questions or concerns or anything that might fuel debate about this.
Thank you very much, Dr. Veraart, for this great talk. And as you said, we now move to the Q&A phase. So I just like to remind everyone to please address your questions using the Q&A box, or just raise your hand. On the meanwhile, Gabriel, do you have any comment or question you would like to share?
Yeah, for sure. Thank you. Thank you very much, Jelle, for this nice talk. So I have two questions that I would like to ask you. So the first is like, if you have tried to validate your action diameter in ex vivo human brain, and if you feel like some adaptation of the model or some tuning will be perhaps necessary to account for tissue fixation or other peculiarities. And my second question is about the spatial resolution. I am seeing that you are using 2 by 2 by 2. I think that was the last study. Do you think we can push it a bit more and we will get some gain, or right now we should stick with 2 by 2 to get decent SNR? Those are my two questions I would like to ask you.
Okay, I'll start with the first one, ex vivo. We know that by doing fixation, we lose diffusivity. So the intrinsic diffusivity drops quite a bit because of the fixation and because of temperature. Now, a lower intrinsic diffusivity is actually beneficial for sensitivity to axon diameter mapping. However, and this is the biggest problem, it also applies to the extraxonal space. So if you want to apply this model on post-mortem tissue, we have to find a way to suppress the extraxonal space. Because of the lower intrinsic diffusivity, we have to go to much longer, much higher B values. And I'm not sure how to achieve that, especially not on scanners like the quantum scanner. We were able to do that on our rat brains, perfusion fixed rat brains, because they had like a 3000 millitesla per meter gradient system where we could push our B values quite a bit. We could play with our individual parameters a lot. But if you're bound to 300 millitesla per meter, I am not sure how easy it will be to fully suppress the extraxonal space before you can actually start probing the intraxonal features only. So that's the main problem. I think maybe bringing up the tissue to body temperature will help in this case, but that's of course like an entire different challenge. The low diffusivity will be a problem in fixation. And then the second question, resolution. I think resolution is always like something we strive for, but what resolution will not change is how the effective radius is encoded. So the effective radius is still weighted. We have that 6th order moment of our axon diameter distribution. going to higher resolution is not going to change that. So the intrinsic bias of those large axons does not depend on the resolution. What depends on the resolution? Signal to noise ratio. So in the end, like if you really keep pushing it, like duration bias will really dominate everything, or non-central chi bias dominate everything, and you might have to think about different ways of reconstructing your data. I mean, which is like work in progress, and we will learn more about it every day. So it might become possible, but there are, I think, challenges at this moment associated with going to larger, higher resolutions, scan time, drop in signal to noise ratio, potential signal biases, without resolving the intrinsic bias to large axons. And if anything, I went the other way. I went from voxels to fiber segments. So I basically went from 1 voxel to about like 100 voxels, lowering my resolution, because I believe that the fiber segment is kind of much more of like a biophysical unit of a building block of our white matter pathways than a single voxel. So maybe in practice, what I would do is like, I mean, it might be a consequence of like acquiring high resolution, but then just averaging more voxels within the segment, which is in the end the same thing because it's independent of the resolution. So I'm not sure that my first efforts would go to higher resolutions without maybe a different specific question in mind. But I think my efforts would go into understanding some of those along tract variabilities that I think remain understood.
Okay, thank you very much.
Thanks. So the next question from Bruce Jenkins. Can you please unmute?
Oh, hi. Yeah, very nice talk. I thought it was very clear and well presented in terms of laying out all the evidence and the reproducibility, et cetera, et cetera. So thank you for that. I just had 2 very simple questions. So the first question is, how much do you have any data on exactly how much shrinkage you get when you're measuring axon diameters in histological preparations, so that you would be able to perhaps, you know, put a bigger error bound on the histological measurements compared to the larger diameters you're measuring with the MRI?
So I'm not sure if it's a simple question. I think there's like lots of research done on like the shrinkage factors, and it varies like from a few percent to like 10%. Depends on like the preservation techniques. Cryopreservation has definitely like helped in like minimizing the shrinkage. But there are actually various things that needs to be considered here. What is the radius of an axon when it's not fully cylindric? Because that's actually like, I think, a biggest step that we take when going from histology to MR. And it's also what I did in my study. We've got like a complex morphological shape, and you basically fit an ellipse on that, on the cross section, and then you take a radius. And even there, the larger radius has to be considered in the end, because that will propagate into the effective MR radius. So I think the main challenge remains interpretation of complex axonal morphology rather than the shrinkage itself, especially the development of like preservation techniques like the cryopreservation. So and I think I've seen his name here, but your former colleague and currently your colleague Hongxi Li has been doing like all kinds of Monte Carlo simulations to basically go from histology to diffusion weighted signal by including the actual morphology. And that is really the way forward, I think, when we can go to electron microscopy, where we have the full morphological model of our axons, put in some random walkers, put in like our actual imaging acquisition metrics, and map to our diffusion weighted signal. Because then we don't have to make assumptions on like circularity of our cross section of the axons. We don't have like the issues with dispersion and stuff like that. So aside from the shrinkage, I think there's like still lots of work to be done to go from histology to MR predictions. And I think the work of Hongxi Li is really like driving this step. So I'm really looking forward to see how this keeps developing.
Yeah, the second question I had very quickly was, in your patient study, I noticed that the percent differences between the controls and the patients was about the same for your measurement of axonal diameter as well as the fractional anisotropy, which of course is a much simpler and easier measurement to make. So there's kind of two facets to that. One is your measurement telling you anything about what you think is actually being measured with a fractional anisotropy at a deeper level? And two, do you want to go for a much more complicated biomarker that is requiring very high gradient strengths to be able to measure, as opposed to something as simple as FA?
I will never question the relevance and the need for like fast and robust biomarkers like FA, but they're intrinsically like sensitive and not specific. And I think especially like in clinical research, that specificity is something that we have to keep like striving for. And if we can decompose FA into like the different cellular processes and we have an increased specificity, I think that's like a major step forward in monitoring disease and progression and therapeutics. So I don't think it will replace FA. I think like a fast diagnostic marker is needed, but it needs to be complemented, especially in certain contexts, with something more specific. And I do believe that high B, like strong diffusion weighted imaging, which is possible on clinical systems. I mean, if we talk about axon diameter mapping, we go to extremes. We go to like those B30,000s and stuff like that. But clinical scanners are able to go to regimes where axons are extremely dominant. And we can already complement our FA with like more axon forward metrics that would allow us to monitor certain processes in more specific terms. So yeah, I do see the value of complementing FA with something more specific without acknowledging the importance of something robust, quick, and sensitive like FA is. And yeah, I think it complements each other.
Thank you very much. So I think we're going to conclude with that. And I'm going to thank you again, Dr. Veraart, for this great talk, and everyone for joining. And we hope to see you next time in person.